Gene therapy is a process in which the manipulation of an organism's genes can be extended to humans. It's an accepted therapy that is used to treat generalized disorders such as cardiovascular disease and cancer, as well as cure inborn errors of metabolism. Liposomes are microscopic beads of lipids specially prepared to enclose the normal gene, and genetically modified viruses can also be used to ferry a normal gene into the body. Two kinds of gene therapy are ex vivo, which is when a cell is removed from the body and then a gene is inserted and then it is returned to the body, and in vivo, when a gene is delivered directly into the body.
Braden's bone marrow extraction went very well, but he's going to be in some pain for a while. Hey, you did great, Braden. We're almost there. Cool.
Can you explain it to us one more time?
Yeah, because it still sounds like you're going to give our son HIV. No, it's gene therapy. You know, Braden lacks the ADA enzyme. Without it, he has no immunity. So in order to get it into him, I'm going to deliver it on a fast replicating virus.
The HIV virus?
Yes, which will disseminate the enzyme until it grows on its own. You want to inject him with HIV? No, not per se. For the past 2 weeks, I've been working to genetically disable an HIV virus. Okay, the HIV is like a hat, okay? The virus is what matters. I've taken off the HIV hat, and I'm going to make it wear an enzyme hat. I know it sounds counterintuitive and scary, but I think this is his best shot. We trust you.
We're gonna go, champ. You gonna be okay?
He's probably going to sleep for the rest of the night.
Sweet dreams, Brady Bug.
He dream about replicating genes.
Or bunnies.
This is how ex vivo gene therapy works. Modified DNA is injected into the vector, which is that green hexagon. So the vector binds to the cell membrane and is then packaged inside a vesicle. That vesicle breaks down, releasing the vector, and the vector injects new genes into the nucleus. The cell then makes protein using the new gene.
As previously mentioned, in vivo gene therapy is when genes are delivered directly into the body. The site of gene delivery correlates to the treatment of different diseases. For example, genes transferred to the brain by injection help treat brain tumors and Huntington, Alzheimer's, and Parkinson's diseases. In vivo therapy in the lungs by aerosol spray treats cystic fibrosis and hereditary emphysema, and in vivo therapy through bone marrow transplant treats sickle cell disease.
Genomics is the study of genomes or a complete genetic makeup and also that of other organisms.
The Human Genome Project, the most well-known genomics endeavor, was a 13-year international project to map all base pairs in the human genome. This project led to structural genomics, or the study of the sequence of DNA base pairs. Repetitive elements occur when a sequence of 2 or more nucleotides is repeated many times along the length of 1 or more chromosomes. Transposons are specific DNA sequences that have the ability to move in and between chromosomes, sometimes altering neighboring genes and decreasing their expression, much like a regulator gene. Functional genomics, where understanding the exact role of a genome in organisms, uses DNA microarrays to show which genes are turned on in a specific organism or cell at particular times and environmental circumstances.
Proteomics is the study of structure, function, and interaction of cellular proteins.
One goal of proteomics is to identify and determine the function of proteins within a particular cell type. Each cell produces thousands of different proteins that can vary not only between cells, but also within the same cell. The study of protein function is viewed as essential to the discovery of new and better drugs, and is why proteomics is a critical field to explore in the future.
And bioinformatics is the application of computer technologies, specifically developed software, and statistical techniques to the study of biological information.
Functional genomics and proteomics depend on computer analysis to find significant patterns in raw data. Bioinformatics is extremely important in human genetics, making it possible to know how genes and proteins interact within cells, determine the function of DNA sequences, and compare the genomes of different organisms.